Welcome back, students. So in this video of microbial concepts, we are going to discuss some interview questions related to food microbiology. Now in this video, I have listed a range of questions starting from say basic to advanced level, but these all 20 questions are just to give you an idea that what type of questions you may get in your interview. Okay, so it is not that if you learn all these 20 questions and if you go to interview that then you will be able to crack your interview. Okay, you need to study thorough. Let it be a confectionery industry or a dairy industry or any meat processing industry. These are the basic questions. Okay, I am again focusing on the term that these are some basic or common questions that you may get. Okay, now the question may vary or a question may get specific depending on what type of industry you are having your interview. Okay, so let's start. So very first and basic question that the interviewer will throw at you is what is microbiology or what is food microbiology? Then why food microbiology is important, or what is the role of microbiologist in QC, or what is the role of microbiologist in food microbiology industry. Okay, food industry. So this is how the interviewer will start taking your interview, and depending on your answer you will get cross questions. Okay, counter questions, and that is how the interviewer will analyze you. Okay, so be confident, be calm, and answer in simple words and in try to answer in 1 to 2 sentences. Okay, so let's start. So very first question is what is food microbiology? So it is study of microorganisms and their interactions in food. Then why it is important? So it is important because food microbiology helps us to ensure food safety by identifying and controlling some harmful pathogens or organisms which are growing in our food sample. Okay, then understanding the spoilage mechanisms, and thus on that we can develop some effective preservative or preservation techniques. Okay, so that is the reason food microbiology is important. Then what is the difference between pathogen or spoilage organism? This can be a question, or simply you can get a question like can you define pathogen and spoilage organisms? Okay, or you can just get a simple question that um, can you define pathogen? Okay, so you can answer like a pathogen refers to a microorganism that can be say bacteria, fungi, or viruses that can cause illness on consuming a contaminated food. Okay, that means that particular pathogen is growing in that contaminated food, and if someone consumes that, then he or she may get uh, or he or she may fall ill. Okay, then spoilage microorganisms, they what they do, so they are the ones which degrade the quality of our food item by appearance. Okay, and the sensory attributes of our food also gets affected. Now what are those sensory attributes? Sensory attributes are the texture, the smell, the taste of a food item. Okay, and spoilage organisms, um, they grow and they affect the quality without necessarily causing the illness. Okay, the next is what is the concept of microbial growth and how it relates to the food spoilage. Now, microbial growth refers to the increase in the population of microorganisms in a given environment. Now, in a given environment here means any food item, for example, raw meat or raw milk, and anything which is related to the industry where you are having your interview. Now, in the context of food spoilage, microbial growth, it leads to the proliferation of spoilage microorganisms. Okay, microbial growth means they are growing, they are proliferating. Okay, uh, which spoilage microorganisms which produce some enzymes, some metabolites, and some waste products that can result in undesirable changes in the sensory qualities. So, what are those sensory qualities we have seen? That is appearance, texture, then smell, taste of your food items. Okay, and thus making it unfit for human consumption. Now, what are some common food-borne pathogens and their associated illnesses? So, try to at least give 3 to 4 examples. So, very easy to remember. I have listed some here. Salmonella causes salmonellosis. Then Escherichia coli, we term it in short E. coli infections, okay, as Escherichia coli stands for E. coli. Then Campylobacter is Campylobacterosis. Listeria monocytogenes causes listeriosis. So these pathogens, they cause some gastrointestinal symptoms. Then you may also get fever, and in severe cases it can also lead to complications and even death. Now, what is the mechanism by which microorganisms can contaminate food? Now, here the mechanisms, the term actually means what are the ways through which microorganisms can enter and contaminate the food item. So, microorganisms can contaminate through mechanisms or ways like direct contact with the contaminated surface or equipments during processing or handling, or through or while packaging also. Then through contaminated water or air, and via vectors like pests or humans which are carrying microorganisms. Okay. Then they can also be introduced through raw ingredients or cross contamination from other contaminated food sources. Okay. So next is, what are some methods which are used to detect and enumerate microorganisms in food sample? Now this question can get a little bit specific here. Like if you are having interview in say um, dairy industry, then you may get that how will you detect and enumerate microorganisms in yogurt. If you are having interview in say meat processing company, then you will get question like list some methods used to detect and enumerate microorganisms in a raw meat. Okay, so you have to answer likewise that the methods include some traditional culture based techniques like streaking, pouring for standard plate count, then membrane filtration techniques, most probable number, even direct microscopic examination, and some biochemical test. Okay, other than that, nowadays there are some molecular methods which are developed and which are reliable in case of large scale food sample testing, like PCR samples, PCR reaction, then DNA sequencing, which enables the detection and identification of a specific microorganism. Okay. Then next is, what is the importance of food preservation, right? So food preservation is very important to prevent food spoilage. This is the basic short answer that you can give. But why? Okay, what is the importance? Why to do preservation? Because it maintains the food quality. It also extends the shelf life, and it ensures that the food is safe for human consumption. How? By inhibiting the growth of microorganisms, the enzymes they produce, and by inhibiting the chemical reactions of these spoilage microorganisms. Right? Then, what is the role of pH in food preservation and microbial growth? Now, some food products, they are at neutral pH, some are at acidic, some are at alkaline. Okay, now that depends, but this is a very basic question on a common basis, so you will just answer like pH plays a very important role in food preservation, as it can directly influence microbial growth, either by promoting them, okay, or either by inhibiting them. Now most of the times we have seen that lowering the pH of food items, that means lower the pH means as in acidic conditions, microbial growth gets inhibited. And when the pH levels are higher, that means alkaline conditions, that means these conditions, they will facilitate the growth and survival of microorganisms. Okay. Then what is water activity and its significance in food microbiology? Now, water activity is a measurement for availability of water for microbial growth and chemical reactions in food. Okay. Now, simply you can remember it as Where there is water, you will find microbial growth. Okay, so lower the water activity, the lower the food spoilage rate. Okay, so it is crucial parameter in food microbiology, as microorganisms they require water to grow, and water activity helps predict the potential of microbial growth, spoilage, and stability of the food products. That's why water activity is also tested in food industries. Then what is the role of microorganisms in the production of fermented foods? Okay, so microorganisms they play a very vital role in production of fermented foods by converting carbohydrates, that means sugars, into other compounds. Okay, like it can be organic acid, it can be alcohol, it can be gases, etc. Now these end products they contribute to the flavor, texture, and preservation of food through acidification. Okay, organic acid means acidification, alcohol means alcohol production, and inhibition of spoilage organisms. Okay, so that's the role why microorganisms are important in fermented foods. Now we use them for production of fermented foods, but it is not the case that we use a microorganism to produce a fermented food so it will not get spoiled by other microorganisms. Okay, there are ways through which it will get spoiled when it comes in contact with, say, air, when it comes with contact with, say, a little bit of water. Okay, so you may get a lot of cross questions on this particular topic as well, that is fermented foods. Then what are some common foodborne toxins which are produced by microorganisms? So you can answer it like some of the foodborne toxins are, for example, botulinum toxin, which is produced by Clostridium botulinum, then aflatoxin produced by Aspergillus flavus, and staphylococcal enterotoxin produced by Staphylococcus aureus. A detailed video is uploaded on aflatoxin on my channel, so you can check it out. The next is what is hazard analysis critical control points. So, and what are their applications in food safety? So, HACCP, it is a systemic approach which is used to identify and assess and control food safety hazards, okay, throughout the production process. So this is the system which will identify and assess and control food safety hazards. Okay, that means there are um, chances that some hazards may happen and that may affect the quality of final products. So that is the reason HACCP system is followed. It involves identifying critical points, critical control points, then establishing control measures, monitoring, and taking some corrective actions to ensure food safety. Okay, so even this is very important point, and if you have done a industrial visit in your say masters, then you will be, yes, you will get a lot of questions on this. Then what is antimicrobial resistance and its implications on food safety? Now antimicrobial resistance is not just a topic um, related to medical microbiology. Okay, why? Because food is also one of important source through which such infections which are antimicrobial resistance they can spread, and it can cause potential risk, okay, to mankind. So antimicrobial resistance, it refers to, what is antimicrobial resistance? So it refers to the ability of microorganisms to withstand the effect of antimicrobial agents, okay, like antibiotics, and make them less susceptible to the treatment. That means the treatment will not work if the organism has antimicrobial resistance. That means it is resistant to the given antibiotics. Then in the context of food safety, it possesses a significant concern and it can lead to the proliferation of drug-resistant pathogens in food chain, increasing the risk and spread of foodborne infections, and reducing the effectiveness of antimicrobial treatments. Okay, then what are some emerging trends and technologies in food microbiology? So, if you are having interview in any kind of food microbiology related industry, then this is what a very important question that you will be getting, that what are the emerging trends and technologies in food microbiology. So a very short but a very good example is you read some recent research articles which are related to the food microbiology technologies. Okay, so you will get or you will be updated on this topic. So some trends and technologies include use of metagenomics for microbial community analysis, then application of nanotechnology for antimicrobial packaging, and the development of rapid detection methods such as biosensors and molecular techniques for efficient and timely detection of pathogens. So if you are not aware about this term, what is biosensors, or you are not having the concept clear, then you can watch a video which is uploaded on my channel. Okay, then what are the challenges and strategies for controlling microbial contamination in food? Then food processing facilities. Then the challenges are maintaining proper sanitation practices, preventing cross contamination, ensuring compliance with food safety regulation. These are the challenges. Okay, then what are the strategies that you can apply? So they include implementation of robust sanitation procedures, training staff on personal hygiene practices, then conducting regular testing and monitoring of food samples, and implementing HACCP systems to identify and control potential hazards. Then next is what are biofilms and what are their significance in food safety. Now biofilms is again a very vast topic, but yes, 2 3 videos are uploaded on my channel, but we are not going to discuss biofilms here. So in short, biofilms, they are communities of microorganisms which adhere to a surface and they form a film, okay, on that particular surface, and they form a film that is so protective because they produce a exopolysaccharide which shields it from any other chemical or say any other agents, okay, that can kill that particular microorganism. So that is biofilm. So in food safety, biofilms can contaminate equipments, okay, and surfaces of food processing facilities. For example, if uh, there is a bioreactor which is being used on a daily basis, and if it is not cleaned properly, then you will find a thin biofilm growing on the surface of that bioreactor. Why? Because there is availability of water, proper temperature, proper pH, then all the nutrients which are required for that particular microorganism to grow and form a biofilm. Okay, now serving as a reservoir of pathogens and then leading to persistent contamination challenges and increasing the risk of cross contamination and foodborne illnesses. Okay, so that's the reason why you need to study biofilms, and then you need to identify them and try to prevent or try to inhibit biofilm formation. Then what are the factors that influence the growth and survival of microorganism in food? So again, factors influencing microbial growth. This video is uploaded on my channel, so you can check that. So the growth and survival of microorganisms in a food item are influenced by factors such as temperature, pH level, very important, water activity, nutrient availability, oxygen levels, presence of preservatives or any antimicrobial agents, which collectively affects the rate of microbial proliferation, then spoilage, and then potential pathogenicity. Okay, then what is the role of preservatives and additives in preventing microbial growth in food? Now, preservatives and additives, they play a role in preventing microbial growth in food by inhibiting the growth of microorganisms and their activity. By activity, I mean production of some enzymes, some metabolites, or some other biochemical reactions. Then extending their shelf life, maintaining product safety and quality, etc. Okay, so they can act as antimicrobial agents or antioxidants or pH regulators to prevent the spoilage and growth of pathogens. Now, whether it will act as antimicrobial agent or antioxidant, this depends on what type of preservative or what type of additive you are adding. Then next and last question is, what is cross contamination and what are the strategies to prevent it? So cross contamination refers to the transfer of harmful microorganisms from one surface or one food item to another and potentially leading to foodborne illnesses. Now, some of the common strategies that one can apply to prevent cross contamination is practicing proper hygiene. Okay, now self hygiene and even your uh, area, working area hygiene or sanitation. Okay, then implementing separate work areas for raw and processed or cooked food, then using separate utensils or say bioreactors and fermenters, and maintaining proper cleaning and sanitation protocols for your food washing and food processing areas. Okay, now this depends on the type of industry you are working in. Then I hope all these questions, which are around 20 questions, so I hope these questions are useful to you. You have an idea now that what um, range of questions you will be getting, so study thoroughly. And do like my videos, do share these videos with your friends, and do subscribe to my channel. Thank you.